Chapter 8, Section 5, Pepto-Glycan Biosynthesis. All right, as the cells are growing, as the cells are dividing, even cells that aren't actively growing and dividing yet just have to sit there and replenish, have to synthesize new cell wall because cell wall is constantly getting damaged. So, what we know happens when we've studied this is that existing peptidoglycan layer that is nice and structurally sound has to be broken, severed, because you can't have this continuous thing and be able to insert new. So you got to break it, insert in new, break it again, insert new, break it again. So we'll see that in rods like E. coli, Klebsiella, whatever, that's going to occur in multiple places where that breaking and repairing, break, repair, as you're trying to increase or have some form of a turnover. Because remember, as I said, things get damaged all the time. So, what do you do? What do you do? What do you do? Well, if you're, a bacilli, you do this at multiple places. If you're a cocci, you're going to start at the quote-unquote equator of the cell where the fifth Z ring is going to go, and then you build out from there. What's interesting is all new peptidoglycan that's going to become part of the cell wall has to be made in the cytoplasm. It's where the enzymes are. It's where the sugars are and all the components. Once it's made, it's going to be transported outside the cell and then assembled. So when you look at a gram-positive bacteria, remember I just said, you have the fifth Z ring there that's kind of acting as an equator. You're going to have these wall bands, these areas where the existing peptidoglycan cell wall is going to be broken. And then you're going to fill in. This is the new growth area. You can actually see this. Here's some high magnification electron microscope pictures. You can see where the cell wall was broken, and then anything in between right here, that's going to be new growth, new peptidoglycan that has been made. Fifth Z seems to be the place. Everything's going to be centered around. Okay, Staphylococcus aureus. Okay, Staphylococcus. Cluster of cocci. So, all right, that means we're going to have fifth Z ring. You can see the orange ring there. And this is where the breaking and the making of new will occur. The fifth Z mutant, well, fifth Z doesn't accumulate in a singular place, which means you get these weirdly shaped daughter cells. These resulting cells, none of them are truly even up close to approximating coccus. The growth just seems to distort it out, this one long cell, as you see there. So for any type of growth or any turnover to occur, you have to have a controlled cutting. Go through and cut certain places, excise certain places that you can then insert into. Bactoprenol, okay? It is a C555 alcohol. So it's a 55 carbon strand alcohol. You take the bactoprenol and then you bond it to N-acetylglucosamine, NAG. NAG will then bind to NAM, and a pentapeptide. So you've got this 55 carbon alcohol and NAG and NAM attached to it. Kind of cool. But remember, this is still in the cytoplasm. You're going to need a transporter. Well, guess what? You got one. And it's called flipase, of all things. Flipase, well, it's going to flip out lipid 2 from cytoplasm, periplasm. So here we go. The hydrophobic portion. 1, 2, 3, 4, 5. This times 9. 1, 2, 3, 4, 5. So there's 45, and you add up the rest. There's your 55 carbons. So it's a little deceptive, the picture, because it's hard to see that right there. And let me make that a little brighter. Oh, where'd my cursor go? There it is. Right there. That 9 times the 5 that are in there, the parentheses, gives you the 45, and you got 5 and 5. So there's your C55. So this is an extremely long hydrophobic portion. Okay. It's going to pretty much, this is going to insert into the membrane. Flipase is then going to flip it around. So this was in the cytoplasm, now in the periplasm. Or if it's a gram-positive, it was in the cytoplasm, now it's in that region between the cytoplasmic membrane and where the Pepto-Glycan cell wall begins. Outside, once it's been flipped out, you get what are referred to as transglycosylases. Transglycosylases is going to remove the C55, the lipid 2 portion. And then they're going to take the NAG and NAM, and they're going to start inserting it into the areas that have been cut or damaged. Autolysins are what are going to make the gaps. That new NAG and NAM can be inserted into. Or if there's going to have to be some form of remodeling because of some environmental signals. Autolysins are going to go through cut, shear off old, damaged, add in new, and all of that. You're going to see, because of the way this lays down, there's going to be wall bands. So it's not a homogeneous, all 360 degrees of the sphere. It's going to be in certain regions as bands. A little band here, a little band there, another band there. So this way it is not spread out, but it is concentrated. By keeping this as tightly controlled as it is, this nice balancing act, you avoid, if you're a cell, a bacterial cell, you avoid autolysis. The autolysins going through and cutting, weakening the cell wall, and then you're immediately inserting new NAG and NAB and increasing, the diameter and the thickness of the peptidoglycan. Well, if there's no peptidoglycan coming out because your flipase isn't working or whatever, or the transglycosylases aren't cutting and removing, well, nothing's stopping the autolysins. They'll keep cutting and cutting and cutting until it's completely weakened the cell wall enough that the cell just pops. So you've got this long lipid 2 thing that they've drawn here that will insert and basically cross, the lipid bilayer. Flipase will then flip it out into this space between the cytoplasmic membrane and the beginning of of the wall. Autolysins are going in, cutting, and then transpeptidases are coming in and joining tetrapeptide, tetrapeptide. Here's a NAG-NAM strand, Pepto-Glycan strand. Here's the other, and there's the cross-link, that bridge across to help strengthen everything. New layers are being laid down. There'll be a new layer. Well, let me change back. A new layer right here, and then a new layer. So this will be new when things are being made. And then out here's old, which will be shed because it's going to be exposed to the environment. So we have insertion of new, so we can make the sphere a little bit more bigger, because remember, it's going to want to divide at some point in time. And then we've got, as I've been referring to, you have the damaged old on the outer surface. So you're going to build new, and then, it's just going to be new, 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 and then, the old stuff's being shed. I've already talked about this. Gram-negative cross-link form between the DAP, on one, and the D-alanine and the other. Our friend Fitz-I, transpeptidase. This aids in, for E. coli, it aids in making that tetrapeptide formation. What's interesting is this transpeptidation, making that peptide-peptide bond between the Pepto-Glycan strands of NAG and NAM. That is the actual site of activity for the antibiotic penicillin. Penicillin makes it so that those two tetrapeptides can't be linked together. Thus, you still have the NAG-NAM strands, but now they're wobbly. They don't have that cross-linking. Every other, NAG-NAM cross-link that's going on. So they become wobbly. Wobbly is not structurally sound, which means any osmotic pressure, especially when there's nothing stopping the autolysin, which is still cutting because, well, it was made and it's going to keep cutting to insert new peptidoglycans, which are all wobbly. Morphology-related proteins and peptidoglycans. So you're going to get quite a few of these penicillin binding proteins. Again, remember, their job isn't to bind penicillin, just penicillin seems to interact with them, binds to it, interacts. These proteins are either acting as transpeptidases or transglycosylases. They're either making the peptides or they're removing the whole lipid 2 portion and releasing the NAG-NAMs. You're also going to get other SEDS proteins are coming to play, ROD-A, MREB again. And then there's a whole class B of minor proteins that can form peptide crosslinks, but they're not as widely distributed as these class A ones.